Yes, good. Okay, so I think this is where we left the last class, right? So let's start with a quick summary as we did. Where did we start from in the last class? I think immunity only, right?
Yes,
Immune
sir.
system. Very good. So who would like to give a quick summary? Anyone would like to volunteer, or I should choose? Sometimes volunteering is good, and it's not a test. So I will help you. If you'll stuck somewhere, if you'll get stuck somewhere, I'll just help you. Anyone would like to do it?
I will.
Yes, great, Alia. Amazing.
Yeah.
I'm so happy.
Uh,
Yes, come on.
so we started with immunity slash immune system. It's
Mhm.
basically is the overall ability of the body to fight against disease causing organisms conferred
Perfect.
by immune system called immunity. Then there are various different structures, layers, and cells organized in our body to defend against pathogens.
Yes.
Basically, there are two types of immunity: innate and acquired. Acquired. Um, so, in innate immunity, it's non-specific and it present since birth. So,
Yes. An
the
acquired one?
acquired one is
That
developed after birth, like
yes,
it's
right.
yeah. And so there are
Correct.
four barriers in innate immunity.
No, the four before the four barriers, yes. Because
Yeah.
innate immunity, like you said, I'm just adding on to your points. Very good. So I'm just adding on that you said that it is non-specific, right? You are correct. Innate immunity is non-specific. And why it is non-specific? Because before we are born, we don't know of the pathogens that we are going to encounter in the world, right? In the real world. When we are inside our mother's womb, we are protected, right? Organisms are protected inside the mother's womb or inside the egg or the cocoon, right? When they come
Yes,
in the
sir.
real world, they start encountering pathogens, viruses, bacteria. So of course our inbuilt immunity cannot be prepared, it cannot predict and cannot be prepared for pathogens. Okay, but it's not like then it's not efficient, or it's not good, or we don't need it. Because some organisms, as I told you, invertebrates, all the invertebrates, they just have innate immunity, right? They do not have a full-fledged acquired immunity. Why I'm using the word full-fledged is because latest research also gives us a hint that they might have some components which are adaptable. Some part of their immunity is actually adaptable, because they also show responses that differ upon subsequent encounters. But majorly, they don't have the players. The players are absent of adaptive immunity. So innate immunity is all they have, and they do good, right? They live their life. They fight the diseases. They are very happy with the innate immunity. So it means it is very, very usable. It is very useful thing. And then innate immunity is divided into basically four barriers. So yes, continue, Alia.
So one of them is physical barrier, which is skin and mucus. Then there is a physiological barrier, which is acid, HCl, in the stomach, saliva, and tears. Then there's a cellular barrier, which is WBC, leukocytes. The cellular barrier consists of different types of WBCs like neutrophils, monocytes, leukocytes, as well as microphages in tissue.
Yes.
Then last one is cytokine barrier. The infected cells secrete protein in interferon, which
Mhm.
protect non-infected cells from further infection.
Yes.
Um, then we did, then we did acquired immunity, aka humoral
It's also known as
immune
adaptive
deficiency.
immunity.
Yeah,
Yes.
adaptive. Yeah.
Yeah,
Uh,
correct.
this is pathogen specific and it is characteristic by memory. So this
Yes.
means that the primary response of body when it encountered the pathogen for very first time, it produces a response called primary response. Then the subsequent encounter with the same pathogen is faster and in high intensity, and it's called the second response.
Yeah.
Both the
And
primary and
it's very intuitive, right? It's very intuitive to understand it. It's logical. The first response, so it's always like you are playing against a team. Let's say Alia's team wants to have a football match with another team. Now this team is for the first time you are playing with them. So you will not know of their techniques, tactics, what is which player's strength, etc. Right? But if you play with them the next time, you are better prepared. Yes or no? It works for both. Yes. So even like the primary response against any pathogen is low intensity and slow. But the secondary response, remember that, is always high intensity and is faster. So this part we call as the memory part of this adaptive immunity. Yes, Alia, continue.
so both of these responses, primary and secondary, are carried out by two special lymphocytes, B lymphocytes and T lymphocytes, also known as B cells or T cells. So
Yes.
B in this stands for in B lymphocyte stands for bone marrow, and whereas in T lymphocyte T stands for thymus. Um, B lymphocytes produce antibodies, whereas in T lymphocyte it doesn't secrete antibodies, but it helps B cells to produce them. And
That's why T cells are often a group of it, often known as helper T cells, because they are helping, right? Yeah. Yes.
and
And then
then we did that this diagram.
yes, the structure. Very good.
Yeah, yes, sir.
Beautiful, Alia. Thank you so much, and thank you for volunteering. Also, okay. So I take on from here. As it, so we started with B lymphocytes first, and as I told you that the B cells, if I call them B cells from now onwards, you know, right? B lymphocytes are called B cells. So B cells secrete or produced an army. This army is of proteins, and these proteins, thousands, lakhs, crores of proteins are there in our body against every pathogen. They just, it's like you make an army, and you of course you expand your army during a war, but it's always good to keep an army ready before the war. Why is it important to keep the army ready before the war?
In case war happens, you already
In
have that
case,
army.
of course, in case war happens, you are ensured that you are protected. Same idea here. Good, great. So this army of proteins is produced by this B lymphocytes. Now you must be wondering, how can proteins act as an army, right? Now this antibody looks something like this. It is a Y-shaped structure with two long chains, also known as heavy chains, and two small chains, also known as light chains. Okay, so each antibody molecule has four peptide chains. Okay, peptide means basically the chains which give rise to protein. So we represent this structure as H2L2 structure, heavy to light to. Do you remember that? H2L2 structure, this one. Make sense, everyone?
Yes, sir.
Yes.
Now, so the name that we give, so these antibodies is like not the biological name. They are against some bodies. Antibiotics are different from antibodies. One confusion that I've seen in students is that they confuse antibodies with antibiotics. You know what are antibiotics?
The
Can
chemical
someone?
substances.
Antibodies are also chemical substances. Come on.
They can they are produced by other microorganisms, or they chemically synthesized, or and they can kill or inhibit the growth of other microorganisms.
So what do antibodies do? They are also secreted by other organisms. They can also be chemically synthesized.
So they are produced by the organism itself. Antibiotics are obtained from other microorganisms, or they are synthetically made.
Antibodies are also synthetically made nowadays. We don't take antibodies, like for antibodies for COVID. We did not take it from all of them from a human blood. We can synthesize it from different organisms. Look at the word antibiotics and antibodies. What is the difference? Both are anti against something that starts with B. What is the difference between biotic and body?
Biotic means life. Body means a particular organism.
Body means a particular element, a body. So anything, any toxin, any organism, any pathogen or any antigen, you can have antibody against that chemical entity. Antibiotics is a broader term which, so if you look, this is A and this is, sorry, this is 1 and this is 2. So if one secretes antibiotic, okay, it will go and act on B and kill B. Okay. But if one secretes antibody, then this pathogen has to come to one's body and get killed there. So antibody is inside. Antibiotics are mostly secreted and released. I think this is what Nabil was trying to say, but he could not use good words and package it well, right, Nabil?
Yes, sir.
I can take the war to you with antibiotics, but not all organisms can produce antibiotics. Or if you bring war to me in my ground, in my body, I will now use my army, which is in my country, antibodies. Make sense, everyone?
Yes, sir.
Antibodies are protein structures, like I have shown you here, but antibiotics can be all different sorts of chemicals, all proteinaceous, non-proteinaceous, everything. Do you understand the second difference?
Yes, sir.
Yes. Antibodies are a part of immunity. Antibiotics are a part of survival strategy or to reduce competition in evolution. So there's a fungi known as Penicillium notatum. It secretes a chemical called penicillin. So wherever this fungi will grow, it will not allow any bacteria to grow in its vicinity. Why? Because if bacteria will also grow in the same on the same source where fungi is growing, there will be a competition for food, resources, water, nutrition, right?
Yes, sir.
So one way to survive is kill the other one. You know, nature is brutal. So fungi secretes penicillin that kills bacteria so that the nearby environment is all clear for the fungi to grow and utilize. Okay. But antibody is like inside our body, that if a pathogen comes, then we are not releasing antibodies in the atmosphere to kill every bacteria wherever it is present. When the bacteria enters our body, we recognize something is bad, something bad has entered, then we want to kill that bad. Okay, and that antibodies can do. Antibodies are not only for bacteria, it can also be for any antigen, any kind of toxin. You know, there are antivenoms. You know antivenoms? Have you heard of the name antivenom?
Yes, sir. Again, snake bites.
Yes. So when a snake bites, in nature, snakes are venomous. You know the difference between a venom and a poison, people? Just connecting some few dots here and there. Just tell me.
Venom is organic toxin, whereas poison is inorganic.
Poison can also be organic or inorganic, both. You're right. For that matter, there's another difference. Like poison can also affect, like poison affects living cells, right? You know that.
Yes, sir.
So poison, if you touch something, okay, by your hand, by your finger, and if it's poisonous, it will start killing the cells of that region. So some frogs are poisonous, poison dart frog. Okay, if it comes in contact with your cells, it will start killing it. So when any predator eats poison or poison dart frog, it starts dying, or it does not like it. But venom is something which, it has to go inside your system, inside the cell or inside the blood to act. So there are some kind of snake venoms which are pure venoms. You can actually drink that venom, some part of that, like in some quantity, of course. But if you have no cut in your mouth, there is no blood, like it cannot reach your blood directly by getting absorbed. You can, if it comes in contact with your finger or your, not tongue, mostly from there it goes inside, but it's just protein, it can get digested by your stomach and not a problem. So venoms to act has to reach our blood or inside the cell. Poison can start acting from outside as well. Make sense to you?
Yes, sir. It's like antibiotics and antibodies in universe.
Uh, Not exactly, but I'm kind of, I don't know what to say, but you have drawn a parallel, but not so what I'm trying to say is there can be an anti-venom because it's a protein. So what happens is when a snake bites, okay, maybe this will clarify things in your head, Nabil. So when a snake bites, it sends the venom inside your blood, right? That's why it bites. Otherwise, the snake can just spray it on your body, no? If I spray venom on your body, nothing will happen. You can hold venom in your hand, you can put it over your body everywhere. Just given that there should be no cuts on your body. You understand? But you cannot put a poison on your skin. If your skin touches a poison, it will start degrading, dying from there. So venom, when it reaches inside the blood upon a snake's bite, it's a protein. So if you can make an anti-venom, which is basically an antibody against that protein, so something that binds to that venom and does not allow that venom to do whatever it is doing. Is it clear, everyone?
Yes, sir.
Yes, sir.
So those are also kind of antibodies only, the antidotes or the anti-venoms. Okay, very well. So so there are many different kind of antibodies, and biologically we use a word called immunoglobulins or immunoglobin. So the word immuno means related to the immune system, right? And globulins means protein. So these are the proteins that help in your immunity, hence they are called immunoglobins, or in short they are called Ig, capital I, small g. Now we have different type of antibodies. They are basically another name for antibodies. Is it clear?
Yes, sir.
So we
Yes,
have
sir.
IgA, IgM, we have IgE and IgG. These four are the most common ones which are studied. So these are some of the antibodies that humans have: IgA, IgM. So it's also called immunoglobulin A, M, E, and G. You can remember it as MEGA.
Sir, I remember as game.
You remember as game. Yeah, very good. Anything. I remember it as a MEGA. So they are the mega players of your adaptive immunity. That's how I remember it. You can remember it that they orchestrate all the game of immunity, right? Anyway, will work. But just remember these four, okay? Now, the next point is that where are these antibodies found?
Sir, in blood.
Yes. So immunoglobulins are found in blood. So write down antibodies.
Question, sir. You
Yes.
said there's anti-venom, right? And
Yeah.
once if a person takes anti-venom against a particular snake, then if he gets bit by the same snake, will it affect him again or?
It depends, because if the antivenom that you put inside also got used, right, to neutralize the effect of the venom which was already inside. And this is something which you are not producing on your own. We are giving it from outside. It's a synthetic kind of antibody against that particular antigen, which in this case is a venom. So the next time, you might again get the severity, you most of the time, because you have exhausted the antibody, you have used the antibody, right? It is not coming naturally from your system. No, you understand what is the point here?
Yes, sir.
So that's where I will teach you then in the next section, active and passive immunity. Some immunities you actively generate, some are just passed on to you, like antivenom. I gave you that immunity through a syringe. Next time again, if you go through the same problem, I again have to give you some immunity, because you are not actively making it inside your body. Okay, makes sense?
Yes, sir.
Humans are not evolved to survive snake bites, right? Because we and snakes are not living in so close proximities all the time that they are biting us every day and we are, evolving against their bites every day. Do you understand, Nabil?
Sir.
Yeah. The pathogens, yes, we are making antibodies. Anyways, yes. So immunoglobulins are found in blood. And you know what is the word that we use for blood in biology?
Hemo.
Heme or humoral. Okay. So just because immunoglobulins are found in the blood, the immunity, write down, the type of acquired immunity, the type of acquired immunity regulated or driven through antibodies is called as humoral immune response or humoral immunity. You can call it anything. Humoral immune response means the immune response which is coming from the heme, from the blood. So it is also known as blood-based immune response or antibody-mediated immune response. So there are many names: humoral immune response, antibody-mediated immune response, or it is also known as blood-based or blood-mediated immune response. Okay, because it's happening through the antibody. That is one in acquired immunity. That is the first type. So which means there has to be a second type, right?
Yes, sir.
And this humoral immune response is majorly mediated by which? It is mediated by B lymphocytes. Okay. Sorry, or I'm writing B cells from now onwards. Is it clear? Humoral immunity is majorly mediated by B cells because only B cells produce antibodies. Is this part clear?
Yes, sir.
Everyone?
Yes,
Now the
sir.
second, yes, now the second part is called cell mediated, CMI. Okay, the cell mediated immune response or cell mediated immunity. So which means antibodies are not playing the major role and it is not driven by the blood, because sometimes it's not always that all the infections and all the antibody, all the antigens and pathogens will go in the blood only, right? They might also affect the tissues or the organs or the cells, right? Yes or no?
Yes, sir.
Yes. So that's why the second type of acquired immunity is cell mediated immunity. And write down, the cell mediated immunity is, sorry, the cell mediated immunity works through, or you can write it, mediated by T cells. Is it clear? So B cells give rise to humoral immune response or humoral immunity, and T cells give rise to cell mediated immunity. Now what happens basically, how can we see that there is a cell mediated immunity? Have you ever heard of the term transplants, organ transplants?
Yes, sir.
What is organ transplant? Yes.
Transferring an organ, a functional organ, from a donor onto another person whose organ doesn't work properly.
So how will the donor survive then?
Yes, sir.
How will the donor survive then?
If the donor is nearing death, or if the donor is having another, like in the case of kidney, where a person can have two kidneys, or when the person is approaching death,
You are not allowed to take any organ from a person approaching death. That's inhumane. So some organs can be donated without any problem. For example, some tissues are easily donatable, like blood. Blood is a tissue, right? And you can
Yes.
just donate it very easily. Another, then moving from there, other things that can be donated easily is a kidney, because we have one pair of kidney, which is two kidneys, and a person can survive fairly well if both the kidneys are healthy. A person can survive very well on only one kidney, and the reason behind it is that when we are under normal physiological conditions, if we are not having toxins, or we are not drinking regularly alcohol, or we are not abusing drugs like cigarette and other drugs, then our kidneys are supposed to be healthy, right? And when both the kidneys are healthy, they don't work on their 100% efficiency, because that's not required. Both are working suboptimal of their efficiency. Even if you donate one kidney, the other one will take the load and will work quite fine for you. So you can donate it. Also, liver can be donated. Half liver you can donate. Liver, because liver regenerates, right? So the donor can just allow some part of its liver to cut and then put it in the recipient, where it will regenerate a bit. So that's the thing. But how easy is a organ transplant? Is it very easy?
So,
No. What is the problem with transplant? One thing that you must have also heard, one term, if you know the term transplant, is rejection of the
Can be
transplant.
rejected.
Yes, perfect. A body rejects the transplanted tissue, right?
yes, sir.
Yes or no? What do we mean by reject the transplanted tissue? Body did not wanted a kidney. It did want a kidney, right? Body has no functional kidney. So if you are giving a kidney to the body should be more than happy. Why is the body rejecting it? Why the body wants
Because
to die? Yes.
it starts attacking. It start
Attacking?
attacking. Yeah. Sorry.
Attacking what?
Yeah. It starts
The
attacking the
kidney.
organ. Yeah, because
But that's what I'm asking. Why? The body
Yes,
needs a kidney, right?
it doesn't
Why?
recognize it. It thinks it to be some external antigen.
Such a big antigen. Antigen were the size of a kidney. You both are along the right lines. I'm just trying to push you, try to diverge you, because that's where you will find your exactly right answer. Yes, Alia. So when you say attacking, what do you mean by that? It's a kidney, right? Body has had kidneys in the past in itself. So what's the problem when I'm putting a new kidney? It is not a pathogen.
it doesn't recognize it. Maybe.
It doesn't recognize it. So you mean that our body can recognize which is my
Maybe,
kidney
yeah.
and which is, yeah, you're correct. You're correct. So there's something called the property of self recognition. You know that? You are absolutely correct. You and Nabil are absolutely correct in saying that they know that what is self and what is non-self. And anything which is non-self is perceived as a threat by the body. You know, it even applies to sperms entering the female tract, because female tract is not, it does not recognize sperms as its own, right?
Yes, sir.
That's where it tries to kill the sperm all the time. And that's where we need millions and millions of sperms, um, to be able to fertilize the ovum, because almost all of them will get killed most of the time. You know, it's a chance even that some most fit sperms which were traveling the fastest, which has the most energy reserves, which were healthy, best in the shape, reached the ovum and fertilized it. And then after fertilization, when the baby starts growing, even that is perceived as a parasite by the mother's body, right?
Yes, sir.
Yeah. So do you understand now the point of self and non-self? Someone is asking something in the chat. Minashi is writing because the body won't accept a foreign particle. Yes. So here the cells which are coming from a donor's body are not recognized by the recipient's body as its own cell. Now, having said that, it feels it. I do you believe that body can know what is self and what is non-self?
Yes, sir.
Yeah, but don't you think it's a problem? Because what if the body start? What if the body cannot differentiate between self and non-self?
Then it will attack. It will start attacking its own self.
Exactly. It will start attacking its own cells. It's like you made a robot to, protect you against your enemies, and the robot forgot to distinguish or stop distinguishing between enemy, who is enemy and who is you. It can only just recognize, okay, it is a cell, it is a human, just like that, and it starts killing humans. So you are also at risk, right?
Yes, sir.
So that way it becomes a double-edged sword, which it actually is in some cases, where some disorders are called autoimmune disorders. Now, do you understand the word auto and immune? Auto means self, immune means immunity. Disorders means our own immunity starts working against our own tissues. Our own immune cells start killing our own tissue cells. Do you understand, everyone?
Yes, sir.
Yeah. So write down cell mediated immunity. Cell mediated immunity makes a body makes a body able to differentiate. Cell mediated immunity makes the body able to differentiate between the self and the non-self tissues, between the self and the non-self tissues. So cell mediated immunity, next point, cell mediated immunity attacks any non-self tissue in the body or any non-self cell in the body. Okay. So how are we able to do transplantations then? Knowing that, you will ask this question: if this is true, We should never be able to do any transplantation. How can we do a transplantation then? Many transplantations do work, right? It's not like
Yes, sir.
all are rejected. Many do work and people are living, surviving with that transplantation. Yes, tell me, how does it work? Why do we say donate organs? If we know that our organ is going to get into someone else's body and get attacked, why bother to donate it? Which means it is working. So how? What do you think? How can we
Because
make it
it
work?
matches with another
A lot
person
of it is matching.
without.
Matching. Yes, one good obvious thought process is matching. What is matching? What are we matching here?
Cells, blood, tissue type.
What do you mean by tissue type or cell type or blood type? What is a type? If a person needs a liver, I'm giving my liver. What is there to be matched? It's a
Sir,
liver. It's not like I'm
the,
putting my kidney in place of the liver. Yes, tell me.
sir, first the blood group.
Blood group. So I'm not putting blood, no, I'm putting the organ, right? The blood later on, whenever we donate organs, we remove all the blood from the organ and then we remove it. So it is filled by the recipient's blood. That's not a problem. Because yes, blood matching has to be there, of course. If it is not possible to remove all the blood, we match the blood group as well. You're right. But what is other thing we are matching? We are basically matching the cell surface receptors because all these antigen antibody interactions happen through receptors, right? The cell type, as you said. So every cell has certain receptors on the top of it. My receptors will be slightly different from, you know, your recept identities. That will distinguish both of us as two different individuals. Okay, and not just the matching. There can never be 100% tissue matching or a blood group matching. You understand? Do you
Yes.
understand that? But still we are able to do transplants. And let me not go to transplant, let me come to something very, very basic. Don't you think when we donate blood, donating blood is a routine, right? Anywhere, any surgery, any accident, any medical case, blood is just, you can just give blood to patient if it just matches the blood group. But do you think that there can ever be 100% matching between your and my blood? It's just that we are looking at three parameters: antigen A, antigen B, antigen O, and Rh factor. So there are four parameters that we look for. But there are more than 200 parameters that we can look for in the blood. So it's never going to be possible that all those 200s match, right?
Yes, sir.
Just the major ones, they match. So do you think that every time when a blood is donated or a recipient takes a blood transfusion, the body is going to fight against the blood? Yes or no? What do you think? Every blood donation, every blood transfusion is actually, it involves a reaction happening. Then why do the patient survive? Why doesn't the patient die?
Because sometimes the antibodies, the antigens in the blood don't match.
No, but why does it survive after blood donation? Why doesn't it die all the time? Every time when there is a blood transfusion, there is a kind of reaction happening in the body, right? Every time. You
Yes,
might
sir.
not feel it because it's less, but it happens. No tissue or no organ or no blood coming from a different organism, sorry, a different human, different individual can be accepted 100% in anyone's body, not even blood. But the problem does not occur because how much blood is always donated or always transfused to one person? How much blood does an average person have? Let's talk about males. How much blood does an average male, human male, has in his body? Around 4.5 liters, right?
Yes, sir.
4 to 4.5, 5 liters, right? But how much do we transfuse? Do we ever, have you ever seen anyone putting 1 liter blood in a patient?
No, sir.
It's always very less, right? Not more than, in severe conditions, not more than 500 grams or something. So which means the incoming foreign particle or the incoming foreign tissue is not that great in amount that it can cause a very severe reaction to kill the patient. If yes, and also we do the blood group matching. So we are actually matching the major antigens, and then we are making sure that the amount of the blood is tolerable. Okay. If you don't match the blood group, of course it's going to cause a bigger problem. Hello, am I audible, everyone? Sorry, I am struggling with some power issues here. Am I audible?
Yes, sir.
Yes.
Minashi, Nabil, Wafia, and Madiha, Alia. Okay, let me share the notebook again. Can you see the notebook, everyone?
Yes, sir.
Yes, sir.
Okay, so what were we discussing? What I was telling you? You're talking about, yes, blood transfusion, right? Yes, transplantations. You're telling about transplantations. Now you said that there needs to be tissue matching or blood group matching before doing any transplantation or graft, right? But still, you know that there can never be 100% match. We just have to work with, doable. Like this is more than 80%, 90% match, so I think the patient will survive. But don't you think that even if there is 1% chance that your cell mediated immunity, which is through T cells, it is active, some or the other day it will start rejecting your cells again. Not your, but the transplanted cells again, right? There is a chance, yes.
Yes, sir.
But if the person has no liver or no kidney, then the person will anyways die. So if you have to choose between the cell mediated immunity and survivability, what will you choose?
Survivability.
Yeah. And how can you choose that? You actually suppress the cell mediated immunity by giving immunosuppressants. So people who undergo transplantations of any organ, they have to take immunosuppressants after that. So what immunosuppressants do? They keep the cell mediated immunity through the T cells inactive. Now there is one benefit and of course one loss, a major loss. The benefit is that the person might be able to survive on a donor's liver or kidney. Okay, but the problem is the person is more susceptible to infections now because the cell mediated immunity is no longer there or is suppressed. Do you understand? Yes.
Yes, sir.
But then the good thing is that the person is alive, so it's better. And when the person is alive, the person can take precautions because he or she has undergone transplantation. So you have to take care for the rest of your life. You cannot just go anywhere. If you have a liver transplantation or a kidney transplantation, you cannot just go anywhere and eat roadside street food, or drink water from anywhere. You would not like your system to be flushed with any other pathogen. So you will take proper care, drink boiled water or proper RO water, eat properly hygienic, sanitized food, properly cooked food, etc. etc. Is it clear?
Yes, sir.
Yes, sir.
Yeah. So cell mediated immunity and humoral immunity both are clear to you, everyone? Anyone has a question? Minashi, Madiha?
Clear, sir. No, sir.
Okay, very well. Now, as we were talking about antibodies and things, there are two types of immunity that can be given to a person. One is called active immunity and other is called passive immunity. So just like when I was, you must have studied, and I was also teaching this long before, that there are two type of diffusions, two type of movement, transportation, active transport and passive transport in the cell, right, of molecules. Remember that diffusion is passive transport
Yes,
and
sir.
movement through channels is active transport, right?
Yes, sir.
Similarly, there is something called active immunity versus passive immunity. Anyone would like to think or guess what it is?
Sir, active immunity means what immunity we make by ourselves against some particular pathogens,
Uh-huh.
whereas passive immunity is something that is given to us
Given to us by whom?
artificially, either through syringe or through parent.
Sorry, your voice is breaking, Nabil. Can you repeat
Either
again?
through a syringe or through a parent.
Either through a, come again, through a
Sir,
syringe.
either through injection or through parents.
Okay, okay, I understood. Anyone else? What do you understand by active and passive immunity, or do you agree with what Nabil said? Anyone would like to add something? Yes, no? So majorly what Nabil said is true. So active immunity is the type of immunity that is produced, you know, inside the host body, okay, through And of course, both active and passive immunity works through antibodies. Antibodies are very important for any immunity, but in active immunity, write down, an active immunity, antibodies are produced in the host body. In active immunity, antibodies are produced in the host body. That's our own. We are actively making our own immunity. Now, how can we do that? How can I give an active immunity to someone? Suppose I want to generate active immunity against, remember we were doing this for COVID. In COVID, we are trying to produce active immunity inside people, right?
Yes, sir.
How can we do that? What are we doing? Antibodies
You
are
could weaken COVID cells or COVID proteins inside the body.
yes, right. It's just the difference is that we are not doing it for COVID. We're not taking that risk, but it was done for polio. You remember polio? The first vaccine that was created, very successful vaccine, was against polio, and it was done by Dr. Salk. And Dr. Salk actually did not patent it. He just donated the vaccine for the welfare of humanity free. That's where polio vaccines, that's why polio vaccines are so cheap and helped to eradicate polio. Okay. So what happening is, like Nabil said very correctly, you use the weakened virus, or we call it the heat killed inactive virus, or we also use the word attenuated. We use the attenuated virus or the microbes, and we put it in the normal person's body who does not have any disease. So this immunity cannot be given to a diseased person. First of all, you can only be immune beforehand. You cannot be immune when you are going through that disease, like from outside. Okay, right? Do you understand?
So
Everyone? Yeah. So, but what purpose will it serve? If I, don't you think I'm taking a risk? I'm just heat killing the virus and putting the virus in the body. What's going to happen? Now let's come to the features of adaptive immunity. Remember that the first response is always low intensity and slow, correct? Why is the first response low and low intensity? Because the body is trying to figure out what the pathogen is and how to kill it. Now, when we give a weakened virus, it means we are just giving a dummy, you know, a real looking, but a model. It's like I train you to fight with a dummy gorilla before putting it in a gorilla cage. So you might learn some things through that fake encounter. You understand? It's called a fake encounter. We are trying to generate a fake encounter in the body. Everyone understands? Where whatever amount of time our body wants to take, it can take that. Why it can take that? Because the virus or the bacteria or the pathogen is weak. It's dead. So it's just an empty shell, empty coat. But we can still recognize the virus and take the time to make antibodies against it. So even if it is slow, we are not at any like we are not at any loss. Once we have figured that out, once our body has made antibodies by looking at this dead attenuated virus or pathogen, next time if the real pathogen attacks, if the real disease, if the real infection happens, what will happen? What will happen?
It will be able to fight it.
Of course, exactly, because we already have an army of antibodies ready, and the next time the pathogen comes, we will be easily able to defeat it. It does not mean that the disease will not happen. Okay, people often think that if you have an active immunity, you will not get the disease at all. It is possible that you will not get any symptom, because before the pathogen can take over your body, you can just crush it because you already know what to do. And all that happened because I trained my body with a fake pathogen. That's what training do. Like how are army people trained? Like for a war, do you go to a war to get training? How do soldiers get trained? Do they actually go for a war to get trained?
Sir.
No, they trained in simulation conditions, right? Where the soldiers, they can learn, they can do mistakes, they can take their time. But when they go into the actual war, they are ready. They know which weapon to use, how, and which enemy to kill how. Similarly, our body, our immunity acts. So an active immunity is developed inside the body. That's called where the antibodies are produced in the body by providing the body with half killed, attenuated cells or pathogens. Okay, so what we are injecting in the body is weakened pathogens. Make sense, everyone?
Yes, sir.
Yep.
Yes,
So write
sir.
down inactive immunity. Antibodies are produced inside the host, and weakened pathogens are injected. So this should come first. First, weakened pathogens are injected, and then the immunity is produced. But just I'm writing it in a different order, but you remember. Now, how is passive immunity different? What is passive? The word passive means
Not
passive,
direct.
passive.
Not direct.
Exactly. Not being directly involved. So our body is not directly involved in this immunity. So what happens in passive is sometimes you don't have the weakened pathogens, or you don't have the time to let the body develop its own antibodies inside. Okay. So what we do is we have to give the person ready-made antibodies. This is not possible for every pathogen, because for that we have to know the pathogen. Clearly it was not possible for COVID at all. So with COVID, the only thing we could do was to give active immunity, because it happened as a pandemic for the first time. But now when we know it, after like few years, we might have passive immunity, like preformed antibodies against it. So in passive immunities, what happens is ready-made antibodies are directly injected in the body to protect the body against any foreign pathogen or any foreign particle or toxin. Do you understand?
Yes, sir.
Yes. Remember when I was teaching you reproductive system, I also taught you a small part of called parturition, the birth of the baby, right? And then lactation happens. Remember that? Post parturition, there is lactation,
Yes,
where
sir.
mothers produce colostrum in their body. The human milk is known as colostrum, right? And remember I told you that colostrum provides the newborn baby with some preformed antibodies. And the most important is IgA. Immunoglobulin A is found in high quantity in the mother's milk, human milk. So mothers, when they are feeding their babies, are also providing them with passive immunity. Now these are antibodies which were made in the mother's body, not in the baby's body, but they are passed on to the baby's body to fight certain infections. Make sense?
Yes, sir.
Yes. Write down preformed antibodies are injected in the body of an organism. Okay. So no weakened pathogen, direct antibodies. So you can write an example: colostrum secreted by mother Colostrum secreted by mother during lactation contains IgA, immunoglobulin A. It's an example of a passive immunity passed on very early in life from the mother to the baby to protect the baby. Another thing, you can also give antibodies to the fetus through placenta. Remember placenta?
Yes, sir.
It was the connecting, it was the physical and the functional, structural and the functional link between the mother and the baby. So many things also get transferred from the baby to the mother and from the mother to the baby through the placenta. And so one of these things is another some antibodies. So through colostrum, we know specifically IgA. Through placenta, IgA, and in some cases IgE is also known to be. It can also go IgG. So write down. The fetus also receives some antibodies through placenta from the mother. Receives antibodies through placenta. Okay. Now these two are examples of passive immunity. Now, now you understood active and passive immunity, everyone? Any doubts? No?
Yes, sir.
So the another thing that you will be asked is a difference between vaccination and immunization. When we're giving vaccine, we are immunizing basically, but what is vaccination and how is it different from immunization? Or is it the same, or is it a subpart of the other? Just like if you understood active and passive immunity, you can tell me, and it's not the world knows about vaccination. You know about vaccination. Probably you all have taken vaccines, right? For COVID. Have you taken vaccines for COVID, everyone?
Yes.
Yes, very good. So why have you taken vaccines for COVID? What do you know about it? If you don't know and you were just taken because the government wanted you to take, that's a bad idea. You should know what the vaccines are doing to you, what the vaccines are. So which vaccine have you taken for COVID? Anyone? Would like to name? You remember the name? AstraZeneca. Pfizer is the company. Sorry, Nabil? The AstraZeneca one.
Yes, sir. I took
Okay.
Pfizer.
Okay. And others?
Pfizer.
Pfizer. Okay. The Covishield. You must be knowing the name Covishield or Covaxin or Sputnik, like that. Anyone has taken Covishield?
Yes, sir.
You, Alia?
Yes, sir.
Yeah. So what do you know about your vaccine? What is that vaccine? What are you taking inside your body? What are they injecting inside? Is it? Yeah. Tell me.
Inactivated virus. So it's like active immunity.
Inactivated virus. So are you okay with viruses being injected in your body?
No, it's like active immunity. Like it's weakened pathogen. So
So are you sure that it's a weakened pathogen?
no, like yeah.
So
Basically, so,
COVID was a very novel virus, Coronaviruses we know about, like SARS-CoV-1, which caused SARS, was another coronavirus. But SARS-CoV-2, this virus, we knew nothing about when it came, right? It was just a pandemic. It emerged. It just broke out. Do you think that within 1, 2, 3, how, and I just tell you, in case of COVID, for the first time in the history of mankind, we have achieved the fastest vaccine production. Within 7 months of the outbreak, we had a vaccine, which is a record. In general, with the before COVID, vaccines used to take 2 to 3 years at the fastest, I'm telling you, to come into the market. This just came within 7 months. And many of these vaccines did not even finish all the steps of trial before coming in use, because we desperately needed it. So do you think that they're going to put viruses in you? Because we have now evolved as technology. What do you need to kill a virus? Tell me. So you are right, Alia, you're correct that they were giving us active immunity. Passive immunity was not possible because we did not know what is the antibody against corona, right? So we could not have produced just the antibody and give it inside you. Okay, so it was active immunity. But what does it need? What do you need to kill a virus? You need to recognize the virus. Rest your immune system will do, right? You need to recognize the virus and make an antibody which can bind to some part of the virus. In case of coronavirus, we have spikes. Remember I told you in the last class, not in the last class, but the coronavirus is called coronavirus because the surface looks like this, right? These are the spikes. And we know that for every virus, the coat is made up of protein, and inside that coat, there is a genetic material, which in this case, in the case of COVID, it is RNA. It is a single-stranded RNA virus, if I'm not wrong, yes. And then this is the protein. We call it spike protein. And for COVID, it's different, like it's novel. So if I can recognize the spike protein of the virus, I can pin down the virus. My immune system can pin down the virus. Yes or no? Yeah. So instead of sending the whole virus, you know, the vaccines, for example, Covishield, because I also took Covishield, and actually I decided which vaccine to take given an option. And I thought that Covishield will be better one because in Covishield, there is no virus, you know, there is a RNA. It is a RNA based vaccine. RNA for what? RNA for the spike protein. Okay. RNA for spike protein gene. Do you understand, everyone? Now there is no virus involved. So what happens is this RNA goes inside my body, gets translated into protein, and that protein is the virus protein. So my body will all of a sudden get panic. Oh shit, we have a virus protein in our cells. So probably it's the virus also. If I just show you the tail of the tiger coming out from the bush, you will be equally alert, right? As you will be when you will see the whole tiger. Yes or no?
Yes, sir.
Make sense, everyone? Make sense or not? To figure out or to get prepared against a tiger, you just need to see the tail of the tiger, right? You don't need the whole tiger. You also can do with the dead tiger. But in this case, though we were not sure that how can we attenuate the virus and how much it can be weakened, what if I inject a weak COVID virus and it goes and start causing the disease again, it's a major problem. So instead we chose a safe way. Just take the RNA for the spike protein, the gene from the gene. This RNA will get translated in our body to become spike protein, and we know that it is just one spike, so it will not cause disease, but it will cause our immune response to elicit a full-fledged war. Make sense?
Yes,
Yes, make sense or not? Yes. So in this case, the motive is to make antibodies. Okay. So in vaccination, write down both for vaccination, or we call it as immunization, write down the principle of immunization, the principle of immunization or vaccination. The principle of immunization, vaccination is based on the property of memory formation. The property of memory formation of the immune system. And basically now we are talking about the adaptive immune system. So I'm not going to mention it again and again. So you know, right? So write down what happens in vaccination, like what we did in the case of COVID also. So in vaccination, write down either three things. You can inject RNA for the gene. You can also inject DNA fragment. There are also DNA vaccines. Okay, we use mostly DNA vaccines for animal diseases like for horses and cows and cattle. There are vaccines against certain diseases which are, some of them are DNA, because RNA is a better thing to control, and also RNA can directly go and make proteins in the cell, so we prefer RNA. Okay, sometimes when it's not possible, then we use DNA. So DNA, RNA, or a protein. Okay, protein of pathogen. We can use these three, or we can use the weakened pathogen itself, like it happened in the case of polio. Weakened pathogen we also call is attenuated pathogen. So all these things are injected right now, are injected in the body. So that what are we doing in vaccination? Basically this thing we are trying to generate. So vaccination generates active immunity, correct? Yes or no? Everyone understands?
Yes, sir.
Whereas when we say immunization, it is a broader term. Vaccination is also a type of immunization, but vaccination is an active immunization. Make sense? This is the difference. Active immunization. But immunization can also be passive. But vaccination can never be passive. Make sense?
Yes, sir.
So in immunization, sometimes to save the life of the person, we don't have time. We just discussed snake bites, right? I told you what we do in snake bites. So snake has bitten a person. Now, you know that snake venom. So just on the basis of snake. So, have you heard that they always tell you, if you are bitten by a snake, try to remember what kind of snake it was, or someone should know which snake has bitten you. That helps a lot. Why does it help?
Because you can select what kind of anti-venom to give.
Yes, if you know the snake, then you know what is the venom of the snake like. So there are many different kind of venoms. Okay, so the toxins in the venom could either be cytotoxic. Cyto means, the word cyto in biology means Cytology, cytokinesis, cytosol, cytoplasm. What does the word cyto means?
Cell.
Cell. Perfect. You had to think for this. Come on. Cyto means cell. Cytotoxin means a toxic substance which kills the cell. If it is neurotoxic, neurotoxic venom, it kills the neurons. Basically, it causes paralysis, right? The person will start getting paralysis in the body, and then the person will die. So it can be cytotoxic, myotoxic, neurotoxic, different, or it could be hemotoxic. You know what does the word hemotoxic means?
Toxic to the blood.
Yes. So what will it cause to the blood? It will make the blood clot.
Yes,
Agglutination. Yes. So the blood starts clotting right there in the veins and in the arteries, and the person gets a heart attack or a heart block and dies. Okay. So different kind of venoms are there, and depending on what kind of snake has bitten and what kind of venom is there, the only thing you can do now, you do not have time to produce active immunization, right? You cannot do vaccines here, so you have to inject preformed antibodies. You know what we studied above. You have to do you have to give passive immunity to the person. Okay, so write down in immunization. Can you think of any other case where this can be done? Snake venom bites is one. Any other area where you need to do this? It also needed in the case of tetanus. All of you might have got a tetanus shot, right? Some or the other time in your life.
Yes, sir.
You know, when we do not give vaccines against tetanus that often, but whenever you feel like you have got a wound from a soil or from a metal or from a rusted metal specifically, so these are the areas where tetanus virus, like the microbe, can be found a lot. Okay, so to give immunity against tetanus, we need a rapid response, a fast response. You cannot, you don't have the time to develop immunity after you have, if you are afraid that you might have got tetanus inside you. Okay, anyone knows what causes tetanus? It's a, sorry, I called it virus, it's a bacteria. Anyone know what is the name? What causes?
Bacteria, sir.
Yes, but which bacteria? It's a Clostridium. You know Clostridium family of bacteria, Clostridium tetani. The name, there are many bacteria who are pathogenic, and the name of the disease is named after the bacteria. Like Streptococcus pneumonia causes pneumonia, and similarly Clostridium tetani causes tetanus. Okay, it's a bacterial disease infection, and the spores of this tetanus bacterium are present in the dust, soil, in rusted metal, etc. So when this happens, when you get a wound, it's better to take a tetanus shot quickly. So there what we do is that a quick immune response is required. So we directly inject the preformed antibodies. We know about tetanus bacteria. We also know about what antibodies will work against it. So we keep a concoction of it already preformed and made, just inject it and save the person from the problem, the disorder, the disease, or the infection. Okay, so that is called passive immunization. Is it clear, everyone?
Yes, sir.
Okay. Okay, very well. So nowadays, the last thing that I will do for today is the way we make antibodies or vaccines now is revolutionary. Earlier, we needed bigger organisms like mouse models. Why it used to take more time? Because to make any kind of antibody, you need to generate it in mouse or horse or rabbits, which are closer to human. So many of these antibodies are generated in mouse, horse, monkeys, etc. But nowadays, because of recombinant DNA technology, we can actually just put that fragment of DNA, that gene, whatever you want to make the protein of, into simple systems like yeast, bacteria. Bacteria is a prokaryote, so maybe it might not be able to work for all the genes in a eukaryote like human, but yeast is a eukaryote, and the good thing is yeast is the simplest eukaryote. You understand, everyone?
Yes, sir.
Yeah. So for example, the hepatitis B vaccine, it's produced in yeast for humans. So hepatitis B, hepatitis means liver inflammation. It's a liver disorder that happens in human livers, but the vaccine for that can be produced in yeast. It makes it very easier. We don't need any organism with a liver and a disease condition to make these vaccines. We can just generate it in yeast through yeast specific, what do you call it, technology, like using yeast as a as a as a vector. Okay, everyone has noted everything. Do you have any doubts you want to ask? So I think we are, yes, the time is over. So in the next class we will deal with autoimmune disorders and allergies. Allergies are also something that we have to do, and then I'll take some major diseases like AIDS and cancer. Okay, and with that we will end this disease part of this chapter. And I think another part is also drug abuse, which is there. It's just like educational part to make you aware of what drugs are harmful, why drugs should not be abused and all. But the major disease part, human health and disease, the disease part will be over with AIDS and cancer.